we have learned about action potential in the skeletal muscle, in neuron, in cardiac muscle, about the fast response action potential and the slow response action potential. Now, let us today discuss about the cardiac muscles. You know, the cardiac cells are connected to each other through the intercalated disc, which include a combination of mechanical junctions and electrical connections. The mechanical connections which keep the cell from pulling apart from one another during contraction include the fossa adherens and the desmosomes. The fossa adherens and the desmosomes. Gap junctions between cardiac muscle cells, on the other hand, provide electrical connections between the cells, which allow propagation of action potential throughout the heart. So, the ventricle and the atria, they function as a syncytium. Now, the myocardium has three types of muscle fibers. Muscle fibers which form the contractile unit consist nearly 99.99% of fibers. Muscle fibers which form pacemaker cells and muscle fibers which form the conductive system. Now we come to the organization of the cardiac muscle. The cardiac muscle is a striated muscle. The Z line transects the I band and represents the point of attachment of the thin filaments. The region between two Z lines is the sarcomere. The thin filaments are composed of actin, troponin, and tropomyosin and extend to the A band. The A band is composed of thick filaments, along with some overlap of the thin filaments, same as your skeletal muscle. The thick filaments are composed of myosin and extend from center of the sarcomere towards the Z line. So you can see in this picture, the sarcomere, the part between two Z lines, the actin, the myosin filament, the thick filament, the thin filament. The myosin filaments are formed to a tail-to-tail association of myosin molecules in the center of the sarcomere, followed by a head-to-tail association as the thick filament extends towards the Z lines. The myosin filament is polarized and poised for pulling the actin filament towards the center of the sarcomere. There are several proteins that may contribute to the organization of the thick and thin filaments, including meromyosin and protein C, which appear to serve as a scaffold for organization of the thin filaments. Nebulin extends along the length of the actin filament and may serve as a scaffold for the thin filament. Alpha-actinin anchors the actin filament to the Z lines. Tropomodulin residues at the end of the actin filament are present and regulates the length of the thin filament. These proteins are present both in skeletal as well as the cardiac muscle. Then you know about the titin filament. The thick filaments are tethered to the Z lines by large elastic proteins. These are called the titin. Titin experiments were postulated to tether myosin to Z lines and thus prevent overstretching of sarcomere. But evidence has been found that titin may participate in cell signaling, perhaps by acting as a stretch sensor and thus modulating protein synthesis in response to stress. Length tension analysis of cardiac muscle show a dynamic increase or dramatic increase in passive tension as cardiac muscle is stretched beyond its resting length. Skeletal muscle, in contrast, tolerates a much greater degree of stretch because passive tension increases to comparable level. The reason for this difference between skeletal and cardiac muscle may be due to the possibility that the stretch of skeletal muscle is typically limited by the range of motion of the joint, which in turn is limited by ligaments, connective tissue surrounding the joint. But the heart, on the other hand, appears to rely on the abundance of connective tissue around cardiac muscle cells to prevent overstretching during periods of increased venous return. What happens in cardiac muscle when you are doing severe exercise? There is increase in venous return, which may be fivefold. But the heart is capable of pumping this extra volume of blood into arterial system with only minor changes in the ventricular volume of the heart. That is, the end diastolic volume increases less than 20%. Though the abundance of connective tissue in the heart limits stretch of the heart during these periods of increased venous return, additional regulatory mechanisms help the heart pump the extra blood that it receives. When we will study about the cardiovascular system, we will see this. Conversely, if the heart were to be overstretched, the contractile ability of the cardiac muscle cells would be expected to decrease because of decreased overlap of thick and thin filaments, thereby resulting in insufficient pumping, increased venous pressure, and perhaps pulmonary edema. Now, the sarcoplasmic reticulum is less developed in cardiac muscle as compared to skeletal muscle. The T tubules and the voltage-gated calcium channels are somewhat similar, but T tubules are much better developed in cardiac muscle as compared to the skeletal muscle, because calcium needs to come from the extracellular fluid into the sarcoplasm. Terminal regions of the sarcoplasmic reticulum above the T tubules or lie just below the sarcolemma and play a key role in the elevation of intracellular calcium during the action potential. The mechanism by which an action potential initiates the release of calcium in the heart, however, differs significantly from the skeletal muscle. The heart contains an abundance of mitochondria, with up to 30% the volume of the heart being occupied by these organelles. The high density of mitochondria provides the heart with great oxidative capacity and more so than typically seen in the skeletal muscle. The sarcolemma of the cardiac muscle also contains, we have seen, T-tubules, which are comparable to the skeletal muscle, but T-tubule positioning in cardiac muscle and skeletal muscle are different. T-tubules are positioned at Z lines in mammalian skeletal muscle. They are positioned at the end of I bands. So in cardiac muscle, the T-tubules are positioned at the Z lines, while in mammalian skeletal muscle, they are positioned at the I band. So this is the difference which says between the positioning of the T-tubules in the cardiac and the skeletal muscle. The cardiac muscles also have fewer or less well-developed connection between the sarcoplasmic reticulum and the T-tubule as compared to the skeletal muscle. So here you can see the location of the T-tubule, the cardiac and the skeletal muscle. You can see one is at the Z line and another is at the AI junction. Diameter in the cardiac muscle, it is much more, while in the skeletal muscle, it is less. L-tubules, narrow tubular cistern, while there it is large dilated cistern in the skeletal muscle. Sarcomeric organization is less regular in cardiac muscle, while more organized or more regularly organized in the skeletal muscle. Now we have already discussed this. Let's revise. Adjacent cardiac muscles are joined end to end by specialized structure which is called as the intercalated discs. Within intercalated discs, we can see gap junctions and desmosomes. The heart functions as a syncytium due to presence of these gap junctions, the ventricular syncytium and the atrial syncytium. You can see the pictures of the gap junction and the desmosome. The cardiac muscle, we have already told, there are plenty of mitochondria present in the cardiac muscles, which provides it the oxidative metabolism. Unlike the skeletal muscle, cardiac muscle have to contract in more than one direction. They are not attached with the bones and joints. Cardiac muscles are striated, meaning they will only contract along their long axis. In order to get contraction in two axes, the fibers, they wrap around. The fibers, they wrap around. So there are pacemaker cells which are present in the different parts of the heart, but the heart beats at the rate of the AC note because the rate of the beating of the AC note is much higher as compared to the other places. Now we come to properties of the cardiac muscle. One is electrical property, another is mechanical property. Electrical property, excitability, autorhythmicity, conductivity. Excitability is your bathmotrophic effect. Conductivity is your dromotrophic effect. Now mechanical contractility. This is the ionotropic effect. Then the cardiac muscle, we have seen there is long refractory period. So cardiac muscles, they cannot be titanized or fatigued. There are some specialized tissue in the cardiac muscles which can generate rhythm on their own. This leads to the property of autorhythmicity. Independent of the nerve supply, they can generate their own rhythms. They are self-excitable. Only 1% of the cardiac muscle fibers, they have got this pacemaker activity and can sit on their own rhythm. These are the SA node, AV node, bundle of his Purkinje fibers. You can see in this picture the SA node, the AV node, then you can see the bundle of his and the Purkinje fibers. So, autorhythmic cells do not have a stable resting memory potential. We have discussed about this slow response action potential in the previous class. There is a prepotential out here. You can see in this diagram the prepotential, and there are some funny channels which open up during this prepotential, and the end part of this prepotential is due to opening of a transient type of calcium channels, and the depolarization is due to influx of calcium ions through the L type of calcium channels. The efflux of potassium ions is responsible for the repolarization of the membrane. And again, when the membrane is somewhat hyperpolarized, what happens? Again, there is a funny current which appears, and there is again increase of the membrane potential and generation of another rhythm. The SA node, the rate is 70 to 80 beats per minute. AV node, it is 40 to 60 beats per minute. AV bundle, the Purkinje fibers, they have a rate of 20 to 35 beats per minute. So the SA node has got the fastest rate. You can see from here, and the heart beats at the rate of the SA node. In this picture, you can see that heart rate, what happens with sympathetic stimulation and parasympathetic stimulation. So the heart rate increases with sympathetic stimulation. What happens when there is sympathetic stimulation? That is the fight and the flight response, which involves activation of beta adrenergic receptors on the heart by norepinephrine, released from the sympathetic nerve terminals, or epinephrine released from adrenal medulla into the bloodstream. There is an increase in force of contraction, which is called as positive inotropic effect. There is also an increase in the rate of relaxation accompanying this beta adrenergic stimulation that results in a short contraction. This increase in the rate of muscle relaxation is termed as positive lusitropic effect. The frequency of contraction of the heart also increases with beta adrenergic stimulation and is termed as positive chronotropic effect. Now, what does this sympathetic stimulation do? So, epinephrine and norepinephrine is released, which activate the beta adrenergic receptors on the cardiac muscle cells, which in turn activates adenyl cyclase, increases CAMP, and promotes CAMP-dependent phosphorylation of numerous proteins in cardiac muscle cells. Both voltage-gated L-type of calcium channels, which are responsible for the calcium trigger, and a protein associated with the Sarka pump called phospholamban are phosphorylated by CAMP-dependent protein kinase. The combined actions of these phosphorylation increases the amount of calcium in the sarcoplasmic reticulum. Phosphorylation of sarcolemmal calcium channels results in more trigger calcium entering the cell, and phosphorylation of phospholamban increases the activity of Sarka, thereby allowing the sarcoplasmic reticulum to accumulate more calcium before the calcium is being extruded out of the cell by the 3-sodium 1-calcium antiporter, which is a form of secondary active transport. The net result is that sarcoplasmic reticulum releases more calcium into cytosol during the next action potential, which promotes more actin-myosin interaction, hence increases the force of contraction. The increased activity of Sarka after sympathetic stimulation also results in a shortened contraction because the rapid reaccumulation of calcium by the sarcoplasmic reticulum. This in turn allows the heart to increase the rate of relaxation, that is the positive lusitropic effect. An additional consequence to sympathetic stimulation is an increase in heart rate through a direct effect on the pacemaker cell. So these are the effects that occur in the heart when there is sympathetic stimulation. Now let us see about the excitation, contraction, coupling in the cardiac muscle. So an action potential, when there is a flight and fight response due to a sympathetic stimulation, what happens? The action potential has arrived in the presynaptic terminal, and we know that typical neuromuscular junction is not present in a cardiac muscle. But what happens when the nerve fibers come near the muscle ending? The gap between them decreases, and there are dilations that are seen in the nerve fibers. These are called varicosities, which contain the neurotransmitters. The neurotransmitters are released from the presynaptic terminal into the synaptic cleft, and there are gap junctions present in these areas, which causes rapid transmission of impulses from the presynaptic terminal to the muscle end plate, and this spreads along the sarcolemmal membrane and comes down via the spreads via the T-tubules. In the T-tubules, there are voltage-gated calcium channels. There are voltage-gated calcium channels. So, in response to voltage, this voltage-gated calcium channels, the dihydropyridine receptor channels, what will happen? They will open up and calcium will move into the cell. The ryanodine receptor channels, which are present in the sarcoplasmic reticulum, is a different isoform as that present in the skeletal muscle. In the skeletal muscle, they are mechanosensitive channels. They are mechanosensitive channels. In the cardiac muscles, they are chemosensitive channels. So when calcium enters the chemical, so this will cause opening of the ryanodine receptor channels, and as a result, calcium will pour out from the sarcoplasmic reticulum into the sarcoplasm. So this process is called electrochemical coupling. While in the skeletal muscle, we have seen there is electromechanical coupling. What happens in the skeletal muscle? The ryanodine receptor channels are mechanosensitive. So a conformational change in the dihydropyridine receptor channels causes a mechanical pull in the ryanodine receptor channels, which causes this channel to open, and as a result, calcium is poured in from the sarcoplasmic reticulum into the sarcoplasm. So the mechanism is electrochemical coupling. The excitation contraction coupling in the cardiac muscle is termed as electrochemical coupling involving calcium-induced release of calcium, whereas the excitation contraction coupling in the skeletal muscle is termed as electromechanical coupling involving direct interaction between the dihydropyridine receptor in the T tubules and the ryanodine receptor in the sarcoplasmic reticulum. The basis for this difference in calcium release mechanisms appears to be dependent on the difference in isoform of the channels. There is also difference in the isoforms of the dihydropyridine receptor channels, their expression in the skeletal muscle and the cardiac muscle. Now we come to the mechanism of contraction. Again, this calcium will bind with troponin C. This calcium will bind with troponin C. 4 calcium binds. So the troponin tropomyosin complex, it moves deeper into the groove of the actin filament. The myosin head has got already ATPase activity. It has broken down ATP into ADP and PI and stored the energy. Whenever the active sites of actin filament are free, they will go and bind with the active sites of the actin filament. Whenever they go and bind with the active sites of the actin filament, there will be tilting of the myosin head, that is the far stroke, and this will release the ADP and PI. So the myosin head again gets attached with another ATP. As soon as the myosin head gets attached with ATP, the myosin head loses its affinity for the active sites of the actin filament. So what happens? The myosin head gets detached from the active sites of the actin filament. The myosin head again breaks down ATP into ADP and PI and again attaches with the next site of the active site of the actin filament. Thus the myosin moves along the active sites of the actin filament, pulling the actin filament along with them. So this causes the decrease in the length of the sarcomere and contraction of the muscle. Now, until the calcium level inside the sarcoplasm is decreased, this interaction between the actin and myosin and the contractile process will continue. So the calcium needs to be either pumped back by a form of primary active transport, that is the sarca pump, into the sarcoplasmic reticulum, or move out of the cell by the sodium calcium exchanger, which is a form of secondary active transport. This sarca pump is kept inhibited by a protein called phospholamban. We have already discussed about that. So, during sympathetic stimulation, this phospholamban is phosphorylated, and as a result, it doesn't inhibit the sarca pump anymore. So the sarca pump becomes more active and pumps back calcium into the sarcoplasmic reticulum. Some calcium moves out of the cell by the sodium calcium exchanger. The energy gradient and the electrical gradient for the sodium calcium exchanger is derived from running of the sodium potassium ATPase pump, that is the form of primary active transport. So sodium moves into, so what happens, there is sodium calcium exchange that occurs from the cell, and calcium moves out of the cell in exchange of sodium, which moves into the cell, that is the sodium calcium exchanger, a form of secondary active transport. Now, the different properties of cardiac muscles can be demonstrated by making the heart stop. That is the quiescent heart. This can be done experimentally. Even if you take the heart out, the heart continues to beat at its own rhythm, and with the application of a strenuous ligature, two strenuous ligatures, strenuous ligature 1 and 2, you can demonstrate the property of autorhythmicity of the heart. Now, let us see about the regulation of the force of contraction. The importance of intracellular calcium. The heart is an electrical syncytium with all cardiac muscle cells contracting during a single beat. It is not possible to increase the force of contraction by recruiting more muscle cells, because all the muscle cells have already been recruited. The ventricle acts as a ventricular syncytium, and the atria, it acts as an atrial syncytium. So the cardiac muscles, that is the ventricle and the atria, they function as syncytium. They function as syncytium. Let us again revise what happens with parasympathetic stimulation. When the cholinergic vagal fibers to nodal tissue are stimulated, the membrane becomes hyperpolarized and the slope of the prepotential is decreased, because the acetylcholine released at the nerve endings increases the potassium conductance of nodal tissue. This action is mediated by M2 muscarinic receptors. In the skeletal muscle, we have seen nicotinic acetylcholine gated receptor channels. So here the acetylcholine receptors are muscarinic type, and they are M2 receptor. So whether a neurotransmitter will be excitatory or inhibitory to an organ depends on the receptor that are present in the end organ. So the action is mediated here by M2 muscarinic receptors, which via the beta gamma subunit of the G protein open a special set of potassium channels. The resulting increase of inhibitory current, that is IK acetylcholine-gated receptor channels, slows the depolarization effect of the IH. That is the channels which are called the funny channels, which get activated during hyperpolarization. They get activated during the hyperpolarization. They are termed as funny channels because they produce a current which is a funny current. This IH usually helps the membrane to begin to depolarize, forming the first part of the prepotential. We know that rhythmically discharging cells have a membrane potential that after each impulse declines to the firing level. Thus, this prepotential or pacemaker potential triggers the next response. So, what happens? This potassium channels slows the depolarizing effect of the IA channels. In addition, activation of M2 receptors decreases cyclic adenosine 3-5- monophosphate in the cells, and this slows the opening of calcium channels. So both of these things will decrease the firing rate. So strong vagal stimulation may abolish the discharge of the heart even for some time. So we have discussed about the sympathetic and parasympathetic stimulation. What are the different changes that occur and why? So you put a first tenors ligature. The heart originally was beating at the rate of the SA node. Now what you have done, you have put a ligature between the atria and the SA node, and so what happens? The impulses from the SA node are not reaching the atria. As a result, after some time, the atria, you can see here, there is a period of pause followed by there is another rhythm that is generated, which is called the atrioventricular rhythm. But you can see here, the rate is much slower. Now you put another ligature between the atria and the ventricle. This is the second tenors ligature. What you see, the heart stops for a bit, and after that, the heart starts beating at the rate of the ventricle. This is called the idioventricular rhythm. When we read about the peer interval in ECG, we will see there are different types of heart block: the first degree heart block, second degree heart block, and complete heart block. In complete heart block, we will see that impulses fail to reach from the atria to the ventricle, and so there is a pause in between, and after that, what happens? The ventricle starts beating at its own rhythm. The person may lose his consciousness for some time before the starting of ventricular beating, and this results in Stokes-Adam syndrome, idioventricular rhythm. You have already seen the excitable property of the cardiac muscles. the fast response action potential and the slow response action potential. And the action potential propagates through the conducting tissues of the heart, from the AC node through the different fibers to the AV node, then through the bundle of his Purkinje fibers, it spreads throughout the heart. These are the fast response action potential. First comes the phase 0, which is due to influx of the sodium ions. You can see there is a steep rise. Then comes the brief period of repolarization, that is phase 1, followed due to opening of a set of potassium channels. Then comes phase of plateau. In this phase, there is calcium channel responsible. The calcium channels, L-type calcium channels are responsible for this phase, and the influx of potassium balances the influx of calcium ions. So there is plateau. Then there is that is the phase 2. Then there is phase of repolarization. This is due to closing of the calcium channels and opening of different sets of potassium channels, which are responsible for influx of potassium. And finally, there is returning to the resting membrane potential of minus 90 millivolt. That is the phase 4. So this was the fast response action potential. Now we have already discussed the relation between the action potential and the contractile response, their comparison in the cardiac muscle and in the skeletal muscle. In the skeletal muscle, the contractile response begins 2 milliseconds after the action potential, and the action potential is also short-lived. While in the cardiac muscle, due to presence of gap junctions, the action potential and the contractile response nearly begins at the same time. The cardiac action potential is long. So in this picture, you can again see the comparison of the cardiac and the muscle action potential and the action potential in the skeletal muscle and the contractile response, in which phase they are occurring in the skeletal muscle and in which phase they are occurring in the cardiac muscle. We know that the refractory period in the cardiac muscle is long, because for generation of another action potential, you will need a critical number of sodium channels to open. What happens in the late phase of phase 0? What happens? The sodium channels become inactivated. Until the sodium channels are closed, however, the strength of current to increase, you cannot generate another action potential. You can see in this picture what is happening. This is the total refractory period. This is the absolute refractory period, and this is the relative refractory period. So the refractory period in the cardiac muscle is long. So you cannot tetanize the cardiac muscle, or the cardiac muscles are not fatigued. So this is the comparison of the action potential in a nerve, a skeletal muscle, and a cardiac muscle. So you can see in the neurons and the skeletal muscle, they are short-lived, while in the cardiac muscle, the duration of action potential is much more. Refractory period is due to mainly the inactivation of the sodium channels, and in the cardiac muscle, the refractory period is long. So in the absolute refractory period, However, the strength of the current you increase, you cannot generate an action potential. But in the relative refractory period, what happens? The sodium channels, many of them, they close. So if you increase the strength of the current, you can generate another action potential. This is the relative refractory period. For generation of the action potential in these tissues, you require a critical number of sodium channels opening, and sodium channel opening is voltage and time dependent. So cardiac muscles, we can see they have got long refractory period, that is nearly 250 milliseconds. In the ventricular muscle, during this period, the membrane is absolutely unresponsive to a next stimulus. So you cannot tetanize this muscles. That is, you cannot summate the contraction. Tetanization is complete summation of contraction with no relaxation in between. So the heart gets times to relax between contraction, which prevents the phenomena of fatigue and allows the heart chamber during diastole to feel for blood to be pumped out during the next contraction effectively. So this is a normal cardiogram recorded from a frog or a toad. You can see there is contraction and relaxation, systole and diastole. This is the mechanical activity of the heart. Mechanical activity of the heart. In this picture, you can see the systole is the downstroke and the diastole is the upstroke. This is due to the mechanical adjustment with the lever. In this picture, you can see here, there is an extra contraction. There is an extra contraction because you have given another stimulus during the relative refractory period. So there is an extra contraction that has occurred. So the next impulse generation is delayed. This is the compensatory pause. So extrasystole and compensatory pause. So extrasystole you can see here and compensatory pause. When a stimulus is again applied in the latter two-thirds of the diastole, an extra contraction is observed. This is followed by a compensatory pause. Extrasystole plus compensatory pause nearly is equal to 2 cardiac cycles. You can see again in this picture, this was normal cardiogram. You gave a stimulus during the systole, no extra response, normal. Now you give the second stimulus during the diastole. You can see another contraction is there. This is the extrasystole. But after that, what you can see, there is a compensatory pause. Then followed by again normal cardiogram. So this is the extrasystole and compensatory pause. Long refractory period. You know, the atria and the ventricle, they function as a syncytium, the atrial syncytium and the ventricular syncytium, and they follow all or none law because all the muscle fibers are recruited at a time due to presence of the gap junctions, Excitation contraction coupling we have discussed in the cardiac muscle is nearly similar to the skeletal muscle, but here it is electrochemical coupling, while in the skeletal muscle it is electromechanical coupling. Then we have seen there is abundance of T-tubule in the cardiac muscles and they are very well developed, while the sarcoplasmic reticulum, there is less abundance, and the cardiac muscle, it conducts impulse. You can see here SA node, from there the bundle, interatrial bundles, it comes to the AV node. From there it goes through the AV junctions. The conduction velocity out here is low because of presence of more conducting fibers out here, and then via the bundle of Fibres and then the Purkinje fibers, it spreads throughout the heart. You can see SA node, intranodal pathways, AV node, AV bundle, bundle branches, Purkinje fibers spreads throughout the heart. Property of conductivity. Now the conduction rate varies from different places to places. In the atrial muscle, it is 0.3 meter per second. Intra-atrial pathways, it is 1 meter per second. In the AV node, you can see the conduction velocity grossly decreases to 0.05 meter per second. In the bundle of Fibres, it is 1 meter per second, while in the Purkinje fibers, it is 4 meter per second. In the ventricular muscle, 0.3 to 0.5 meter per second. So the conduction velocity varies from one tissue to the other. It is maximum at the Purkinje fibers and minimum at the AV node. Wherever there is less gap junctions and more fibrous tissues, the conduction velocity decreases. Where there are more gap junctions, less fibrous tissues, the conduction velocity increases. So criteria for state of excitation and efficient cardiac function. Atrial excitation and contraction should be complete before onset of ventricular contraction, which ensures complete filling of ventricle during diastole. Excitation of cardiac muscle fibers should be coordinated. That is, they should function as a syncytium for effective contraction. Smooth uniform contraction is essential to squeeze out the blood. Part of atria, pair of atria and ventricles, they function coordinately as a ventricular syncytium and as a atrial syncytium. So what happens? Which permits synchronized pumping of blood into pulmonary as well as the systemic circulation. So we have discussed about the properties of cardiac muscle, then the properties of the action potential, the excitation, contraction, coupling of the cardiac muscle. All we have discussed. So go back and read them, and next day we will discuss about the smooth.